So it's great to have you, Ferdinand and Lotte. Please take a seat.
Yeah.
Happy to have you. Before we start, I'd like to say a couple of words on why we are doing this. So every year, every edition of our demo day, we have an Alumnus update, right? So when you look at the reason why we are doing that is because there's quite a lot of research about startups and scale-ups. And if you think about it and if you look at the data, we're quite good in starting. So starting is done in the Netherlands, in Europe, relatively well. But scaling is, let's say, where we can really improve on, right? So that's why we have invited Hudson River Biotechnology on stage, but also have the vegetarian butcher later in the program, because we'd like to share these learnings where we are, which we are facing. And it's great to have you, Lotte, and Ferdinand here today with us. Let me start with you, Ferdinand. So back in 2018, you started the company. Was there how did it happen? Was there this technology that was so exciting that you were like, okay, I have to do something with it? Or how did you start the company?
Yeah, so we're a bit unique, I think, compared to many other companies also that we see here, in that we didn't start from, say, a technology spun out from a university or something like that, but we were looking around in the market for questions to solve that were in the realm of things that me and the co-founder worked on. At the same time, the CRISPR technology started to dominate the medical industry, you could say. So yeah, we started working with that, and we actually started with the opportunity that we saw in the plant breeding business in this case, and start to look around for technologies that we could use to help apply CRISPR in plant breeding. Yeah. And from that, yeah, we started with one business case and then grew to what we are now.
And you started with 3 people in Wageningen in a small lab, or how did this?
How did it start? Tiny. It was, it was an idea at the beginning, and it was me and the other co-founder running around talking to people. At some point, we got traction with the ideas, the stories that we had, so we managed to land a grant from the European Union, a small startup grant, and we used that to rent lab space basically at Wageningen University. And then quickly afterwards, we could attract our first employee. Later on, a second employee who is still with us, which is wonderful. And then from there came fundraising, seed funding, and that helped us move into our own lab. We became too big to be a subletter at the university, so to speak. You collect the puzzle pieces and build the story from there. Yeah.
Great, impressive. Hey, Lotte, indeed, Ferdinand already shared, you're an employee number 2.
Yeah.
I think you started as a researcher in the company, or?
Yes, I don't remember how many functions I had from then till now, but indeed I started as a researcher, and actually this was the most surprising to me in the beginning, that there is no IP, what's your technology? You have this idea, but it still seemed very, very exciting and very appealing. So it was a point of my career that I also could say, why not? I wanted to transition out of academia. I was fresh out of academia, but yeah. So,
hey, and as an R&D director, are you able to walk within the strawberries every day, or is it
No. Most of our plants are in little pots in sterile culture. So often we say also, do you have greenhouses? We do not. Sometimes we rent at the university. But most of the clients that we work with have the most economical for them is to facilitate that part, whereas we really apply the technology. That's also what we always say. We are the technology experts. They are the crop experts. And together we collaborate to create something new.
Tell us a little bit more about the platform and the technology itself.
Right. So as I said, so we apply CRISPR. So we take the plants from the client and we break them down into singular cells. And those singular cells, we put the CRISPR machinery inside to do the job that it needs to do. And then from that single cell, we regenerate the plant again. And by doing it in this way, we also are completely transgene free. And also completely, the genetics are stable after that because they all originate from that single cell. And I think that's what the strength that we have also, because we can do this for any plant, so we're not restricted to a plant that we can transform in a way that are how it's generally done in a lot of academic labs or companies. So agnostic, species agnostic.
Great. Yeah. Hey, Ferdinand, when we prepared for this session, you were sharing that I think 50% of your company comes from outside the Netherlands, right? So tell me a little bit, like what is it like to work in a company and to build a company with such a diverse set of people?
Yeah, so we do that by design. Wageningen is a wonderful place, of course, to find talent. With the university as a wonderful pond to fish in. But we wanted to have a much more diverse group of people to work with to make sure we have people from different countries, different cultures, different types of education, different experience all together to leverage what they bring and get to a much bigger, better product and service that way. What it means is that you have to spend more time on the front end, finding the right people. There's visa sponsorship, etc. that can get involved. And in running the company with such a diverse group of people, there's a lot of cultures. There's different languages. There can be sensitivities in communication, so you need to be more patient with each other, have respect and understanding for the other people in your company, and take the time to learn about that. Once that works, however, you start to reap the benefits, and you see the synergistic effect that you get from having such a diverse pool of people.
Yeah, I guess in plant breeding, it's a highly international field, right? So you benefit a lot from that.
Yeah, absolutely.
Lotte, in the video you talked about one of the upsides of being connected to StartLife is also that you see what not to do. So what ideas did you not proceed, or how?
In the beginning, we had a wonderful technology that also helped identifying what to target in the genome. We had a partner in that, and it was a wonderful technology. It tingles all the scientific bubbles. But we noticed that it was too expensive to execute. So it may be later also. It's not the time now. A lot of people wanted to have a proof of concept. They know with the target they want to hit. Worrying about this was more something to worry about in the future. Yeah. And there's been more things that you sometimes think about. There's so much you can do now, like with synthetic biology. It's like, oh, let's AI this and then screen everything. And then you go like, oh, but what would be benefit? And it's like, oh, but you could just buy 10 and test 10 and then you have your... Yeah, you could. But so, and especially the academic mind switch sometimes really needs, because we have a lot of academics and they like get excited very quickly with something new and abandoning maybe existing project and picking up the new one. But cool. There's often we go like, no, let's focus. Yeah.
Okay, are there any questions from the audience, the online audience, or here in person for Hudson? In front?
Yeah, one question. No, gotta wait for the cube.
Is it not GMO?
Right.
Because it is a spacer you insert, right?
It depends on where you ask the question. Yeah. So GMO is
in Europe.
Yeah,
Holland to be precise.
When you ask the question.
Yeah. No, so in Europe, the regulation is still in limbo. So there's been a proposal approved by the Commission on how to handle CRISPR-modified crops. It gets a bit technical, but it depends on what type of edit you make with the CRISPR machinery, and that makes it fall into sort of a light or a heavier category. And the idea is to deregulate the light category and to keep GMO regulations in place for the heavy category. And now we're basically waiting for the next step in the EU regulatory process to get towards approval. The general consensus is that we are moving in that direction, but it will take time to get everyone on board and to negotiate the final language there.
And where will your revenue come from in the meantime?
Well, it is already coming from various places, including Europe, so that's maybe good to know. So a lot of companies are already investing in CRISPR technology, even though you cannot yet sell any CRISPR products here. But so, it's an R&D tool as well. You can use it for diagnostics, analysis, etc. In addition, we have customers, for example, in the United States, in Brazil, South Africa, and there they look differently at the regulatory question.
Thanks.
In the back, I think there's either you poking a red. Yeah.
Hi. So if I understand correctly, you are generating plants from single cell, and I was wondering, you're probably using a lot of regeneration protein like WUSHEL and others to regenerate your plant, and I was wondering how it affects the final stability of the genome in your final product.
Yeah. No, good question. So we always have, especially when tissue culturing, there can always be changes, as you can always get changes always. So we thoroughly screen for that afterwards, and we have a high throughput screening method to test all kinds of media conditions and regeneration boosters, etc. So there's always that possibility, but we have found that mostly it varies from species to species of how bad this is, but we can scale for that, and it is not affected by the editing itself. So it's really the tissue culturing, what we are already used to a lot, of which we are aware sometimes introduces variation. Yeah.
It's not exactly tissue culture, it's generating a plant from a cell. So
yes,
it's more than...
Sure, yeah. It's the more advanced tissue culturing for sure, yes.
Wow, nice stroke in there, right? Yes.
Yeah.
First of all, a remark. It's not an easy choice, strawberry, to start with, especially in genome editing, but maybe you can reflect on that. But my question is, I've been working for a seed company for 36 years, always been on the other side. And how do you deal with always that question concerning, please provide us freedom to operate?
Yeah, so the first question, why strawberry? So it wasn't actually the first crop that we worked on.
Because they taste good, right? Simple.
Yeah, I mean,
they look good.
You actually get the little berries when you grow them in the in vitro pots. Amazing. Yeah. No, so the crops we choose are in big part driven by demand from the market. So obviously we have a few of our test horses in house, like tomato, that we like to work on to test new stuff. But it's mainly questions that we get from customers. And so we at some point started a collaboration with a company that was very interested in doing CRISPR in strawberries. And that was a reason to trigger this. And yeah, we were actually successful in getting that protocol up and running and moving forward. And then on the FTO side, so that's indeed always a difficult question. And it's one that you have to break down into different pieces of FTO. You may be referring specifically to the CRISPR side of it. That's, of course, the difficult part. We use an enzyme called MAD7 from a company called Inscripta. We have a license to use this enzyme. They have their own freedom to operate in this space. And in addition, some of our customers have their own CRISPR licenses, for example, to the Cas9 or the Cas12a enzyme. And so it's a conversation with our customers on which choices we make there. The workflow that we have is independent essentially of which CRISPR enzyme you use. So we like to work with MAD7, but we can equally well work with others. If it's a really new one, we'll have to do some tuning to get our editing percentages up, etc. But yeah, we address that by being flexible and making sure that you have the correct licenses in place to work with the enzymes that we use.
Great. Any last questions from the audience? Otherwise, I'd like to thank you. I have a small present for you, and it relates to a different crop, which I will get from the back.
This is your wish list, Loet.
This is indeed my wish list. So it's a bush of green gold, as we call it, asparagus. It's harvested on Sunday by my dad, and he asked if you can somehow engineer the crop in such a way that they become taller, so he doesn't have to.
Okay. Yeah. Maybe there's easier solutions to that, but we'll look into it.
Okay, thank you.
Thank you so much. It's wonderful.
Thank you well.